Okay, perfect. Let's get started. Hello, I wish you all a happy new year and welcome to the ZOOMinar. I know 8 a.m. is too early for those from the West Coast. My apologies for the inconvenience. I'm sure that they will watch the videos posted on the YouTube. Today we have two excellent speakers, Dr. Per Westermark from Uppsala University and Dr. Marie Doumic. It's my pleasure to introduce the first speaker, Dr. Per Westermark from Uppsala University, Sweden. Per obtained a MD and PhD degrees from Uppsala University and became a faculty at Uppsala in the Department of Immunology, Genetics, and Pathology. For about 10 years, he left to join Linköping University in Sweden and then returned back to Uppsala. He's a pioneer in the field of amyloid, protein aggregation, amyloid fibers, and so on. He has been working on many different amyloid systems, both local and systemic, and has discovered several amyloid fibril proteins. In 1987, his group discovered amylin, or also known as IAPP, islet amyloid polypeptide protein. In 1990, together with his co-workers, he showed that the fibrils in senile systemic amyloidosis is derived from wild-type transthyretin. He has been particularly interested in the improvement of diagnostic methods. And in 1970, he has developed the fine needle aspiration biopsy method for the diagnosis of systemic amyloidosis. He has published more than 450 peer-reviewed publications. He is also the editor of the journal Amyloid. With that introduction, I would like to welcome Dr. Westermark. Per, get started when you're ready.
Thank you very much, Rams, for this. I'm sorry for this delay with my presentation due to I'm not very fond of working with the computers, actually. Anyway, I will talk about TTR amyloidosis. And TTR amyloidosis is a systemic amyloidosis, meaning that the protein or the protein precursor is produced at one site, transported by the plasma, blood plasma, to the sites of depositions that can vary. Kidneys, heart, spleen, and so on are common, and the production places are varying. Also, often the liver can be the bone marrow and so on. And all forms of systemic amyloidosis are difficult to diagnose and are usually diagnosed very late. TTR, transthyretin, is one of the main systemic amyloid proteins. It's produced by the liver as an homotetrameric plasma protein, and each monomer consists of 127 amino acid residues. And the protein forms a tetrameric structure with a channel in the middle, and is stabilized by ligands that are bound in these channels, for example, thyroxine, and also used for treatment by synthetic ligands, tafamidis. And this is a simple picture showing how the liver makes the TTR, synthesizes it, delivers it to the plasma as a tetramer, and for forming amyloid, the dissociation of the tetramer into monomers are necessary, followed by misfolding and aggregation into different kinds of aggregates, including amyloid fibrils that are placed in different organs. This is the same thing shown here by a simplified drawing by Jeff Kelly, and it points to the fact that ligands bound to the tetramer are important for keeping the stability of the protein, and the rate-limiting step for forming amyloidosis is dissociation of the tetrameric protein into monomers, and the monomers are prone to misfold and make different kinds of aggregates. We know the fibrils, and the fibrils are certainly important for the clinical manifestation, but also other pre-amyloid forms, aggregates, are important in different ways. It's not absolutely fully known how they look or how they work. TTR amyloidosis, called ATTR, A for amyloid, is not one disease, but are several diseases, and can be divided up into two main groups. One is hereditary and one is non-hereditary, where the hereditary depends on mutations in the TTR gene, while the wild type, of course, is wild type TTR, and there is no mutation for this disease. And we start with wild type TTR amyloidosis, was earlier called senile systemic amyloidosis. By the name, we understand that it is an age-dependent disease. It's rare below the age of 60, 70, but becomes then quite common, and a number of autopsy studies have shown that in an unselected material that almost a quarter of persons above the age of 80 have some deposits of TTR amyloidosis in the heart. But most have fairly small amounts of deposits, and it's not known if these are important or not. But some get a large amount of amyloid that is increasing, and in those cases, the disease creates a heart disease mainly, that it may be severe and is so mainly in men. Is clinical ATTR amyloidosis of wild type is mainly a male disease. It resembles very much some other systemic amyloidosis of TTR origin that are hereditary as well. I'm coming back to type A and type B soon here. Hereditary ATTR amyloidosis, on the other hand, shows many different mutations, and almost any mutation in the TTR gene creates forms of TTR amyloidosis. They are usually, or almost always, autosomal dominant inherited, and the penetrance may vary quite a lot. And what is also typical in most forms, at least, is that both wild type and mutant TTR are deposited as amyloid. So in some way, it's not only the mutant protein that aggregates. And usually there is polyneuropathy and cardiomyopathy, but in different proportions, depending on patients and depending on the mutations. And it can also occur in varying ages. The most widely spread mutation causing amyloidosis in the TTR gene is with a substitution of methionine at position 30 from valine. And it occurs all over the world, and particularly in Japan, Portugal, and also Sweden, but also in other countries like United States and France and so on. We saw a number of years ago that if we looked at MET30, ATTR valine 30 MET30 is more easy to say, and looked at cardiac specimens in the microscope, polarization microscope, after staining with Congo red. Congo red is the usual way to look at amyloid in tissue sections. We saw that we could see that there were differences between patients. Some had very glittering amyloid deposits, had a strong affinity for Congo red, showed a beautiful birefringence, while other patients had a non-glittering appearance with weak staining with congruence. So we thought that this seemed to be differences. There were not intermediates, but just two very clear groups. And this could also be seen in the electron microscope, that those that had glittering appearance consisted of fibrils that were long, parallel bundles. And they also had a tendency to be intracellular in the heart muscle, while the other one had more haphazard orientation of the fibrils and did not go into the cardiomyocytes. So we call this kind type A and this one type B. And we also saw that there were molecular differences between these two types. The one with glittering appearance consisted of full-length TTR molecules. And while the other type A, had both full-length but particularly fragmented TTR. And these fragments are C-terminal parts of the TTR molecule, starting at usually at 46, 40, 49, and 52, but there are other fragments as well. And so morphologically, we could divide the amyloid into two groups, and that is true for TTR of this kind, not for the other mutations or most other mutations for fully unknown reasons. But so this is what I said already, that there are two different morphological types of ATTR amyloidosis of this origin. And that it was interesting, but of uncertain importance until we also, with collaboration with Ole Sur's group in Umeå, could show that these actually have important clinical importance as well. Now, I think it may be place to say something about what the amyloidogenesis is in TTR amyloidosis, or how the misfolding may behave there. And there have been several suggestions here, and but only a few have been directed to the different types, type A or type B. And for type A, that with fragments, there has been good evidence that mechanoenzymatic cleavage of transthyretin may be important, meaning that shear forces could create misfolding of and cleavage in the monomers in the tetrameric protein that creates dissociation and then formation of amyloid with fairly small changes in the three-dimensional structure of the monomeric protein. And that is, of course, attractive, since TTR amyloidosis has so strong affinity for the heart, where one can believe that there is a strong tendency for shear forces. The problem is that when the studies of cryo-electron microscopy were performed in Marcus Fendrich's group, that showed that the structure of the TTR deposits in type A amyloidosis is very different from the native structure. So this seems to have to include a very strong defolding and refolding of the protein into flat molecules that you can see here. And another thing that was surprising in this cryo-EM is that it seems also to be the N-terminal part of the TTR molecule present, which indicates that you have the cleavage after that the amyloid form has been performed. It's a bit difficult to get these two things together, and I think that the future will show what the real truth is here. And more studies are, of course, important. One thing is important, and that is that this type of defolding and refolding seems also to be true for the wild type form that always has type A fibrils. So type A amyloidosis of MET30 and the wild type are closely related, and are closely related also clinically. So I said that there are differences between these two types of amyloidosis, and we got to understand that after collaborations with the Ole Suhrs group in Umeå, who are taking care of these patients. So those with type A amyloidosis are usually getting their disease at high age, while type B are getting the disease much earlier. Both have polyneuropathy, but in somewhat different ways. And most importantly, type A has a tendency to progressive restrictive cardiomyopathy, with a heart slowly growing and finally takes the patient's life. So type A is a more severe disease in type B in many, at least concerning the heart. The first way to treat TTR amyloidosis of this kind was by liver transplantation. And that has been shown later that this works quite nicely with type B patients, but not with type A, since the progression of the heart, I see a spelling error here, heart disease after liver transplantation. I'm coming back to that quite soon. And this is what it can look like in this restrictive progressive cardiac amyloidosis. I can say that at least 50% of this heart is cut transversely, as you understand, with the left chamber here and the right here. And the interventricular septum is here. And this septum should normally be less than 12 millimeters, but is in this case 25 millimeters. So at least 50% of the material here is amyloid, and with some residual muscle tissue, of course. So it's a terrible disease. As you have here some data on the two phenotypes, A and B, with the glittering, as I said, heart involvement here as measured as interventricular septum thickness, with the thick septum in type A and thinner septum in type B. But more important here, I think, is that if we look at how much wild type there is in the cardiac amyloid of the type of these patients, there is a big difference. And these are from patients that have not been treated in any way. And it can, the two alleles of TTR are both expressed in about the same degree in all patients with ATTR amyloidosis of V30M kind. So one should expect that there should be about 50% of wild type and 50% of the mutated protein in the deposits. And that is true for the B patients who do not get this progressive disease. But as you can see, in the patients with type A amyloidosis, the amyloid consists mainly of wild type TTR of some reason that is not fully understood. So it seems in a way that the amyloid in type A TTR amyloidosis forms by seeding and that recruits molecules, wild type molecules. And that is something that one can see in the microscope as well, that you have a very clear patchy distribution of amyloid. So one can believe that you have a seed here, a nucleus here, and so on. And that is also very much seen in the lung, which is also a site for amyloid deposits in ATTR amyloidosis. You see this is immunohistochemistry. So the brown dots here are amyloid deposits, and these are very discrete and very focal. So then one can ask if these differences can depend on two self-propagating aggregates from transthyretin as strains then, which, is true, has been shown for prion protein, that the same prion protein can yield distinct self-propagation states shown in vitro, but that also can explain the differences between the different strains. And this has also been shown for A beta, actually. And as a matter of fact, Eisenberg's from Eisenberg's group, it has been shown that TTR wild-type monomers can be seeded with ex vivo ATTR amyloid fibrils. I feel fairly sure that they didn't say that it's type A, but it seemed to be type A fibrils. And as if you put, and here shown by thioflavin T assay, with seeds put in here or seeds put in here, you can see an immediate development of fibrils in the thioflavin T assay. And that is also a possible, the strains may be a possible explanation for the findings from Kelly's group, where they looked with a probe that binds to misfolded ATTR amyloidosis in plasma samples from patients of different kinds. And they've showed that patients with this mutation, MET30 mutations, had something circulating in plasma, misfolded protein of TTR origin in the plasma in the polyneuropathy patients that they, most of them at least, that they looked at, and but did not find them at least in wild type cardiomyopathy. So it seems that the misfolded TTR proteins circulating are different between this form that probably is type B and this form that we know is type A. And we have a problem here. We don't have any good yet mouse model for ATTR amyloidosis. And cardiac biopsies are difficult to obtain from human, of course. But it's another way to get samples. And since these are systemic amyloidosis, we can use tissue from other parts of the body. And the most simple part to get is from the subcutaneous tissue. And also in the subcutaneous tissue, you can see the same kind of variability with type A fibrils and type B fibrils and type A fibrils. This can be obtained easily by this way with a punch biopsy, where you can get a good amount of fat tissue and then study in different ways. And also in the subcutaneous fat tissue, you can see typical type B pattern with parallel fibrils, or type A with these short and haphazardly oriented fibrils. And we have used this method to look at amyloid in patients who have been liver transplanted. Liver transplantation has been used as a treatment for 30 years now, and hoped to be stop the development of the disease. But it was shown to halt the disease in type B patients, but not in type A patients. And that depends on recruitment of wild type protein. And here you can see patients that have undergone liver transplantation, and the content of wild type TTR in the deposits in the two different groups. Here, the squares are patients with type A amyloidosis, and you can see that the amount of wild type protein increases within 5 years from something on 70 up to almost 100%. So there is an exchange going on in the fibrils, very obviously. On the other hand, patients with type 2, with type B amyloidosis, they also had a large amount of wild type, but not as much as the type A patients. It's another thing that is very obvious in this picture also, and that is that there are no type A patients after 5 years, but you can see that type B patients occur here to almost 15 years. And that depends on that the type A cardiomyopathy kills these patients. So the prognosis is much better for the type B patient compared to the type A patients. And we could get some interesting information from something called liver domino transplantation. Liver transplantation needs, of course, a liver from a deceased, from a patient that has died of some reason. And there is always a shortage of livers. So therefore, the liver from a recipient or from a patient that is treated by liver transplantation, that liver contains only a small amount of amyloid. It produces wild type and mutant protein, but otherwise it works nicely. So it is often used to give to patients who are severely ill in a liver disease. And this liver is then discarded, of course. And it was believed that these patients would not develop any amyloidosis in at least 20 or 30 years, since the disease does not occur in children, for example. But that was not true. So we have looked at fat tissue in both donors and recipients of these. So the donors had the disease and the recipients were all wild type, but got livers from those with mutations, and that produced the mutant TTR. And what is striking here is that if the fibril type in almost all of the donors had full-length TTR type B fibrils, and those who developed amyloidosis in the recipients, which actually happened sometimes already after 3 years, much earlier than was believed. The recipients also had full length. We had only one with a donor with fragmented, and that patient did not develop amyloidosis. We can't say anything about that. But it's very obvious that all full-length donors created full-length amyloidosis in type B amyloidosis in the recipients. And then we can question, does it depend on seeding? And there was one more information that we got from this, and that is that this amyloid had the distribution of type B amyloidosis. So I think we have fairly strong evidence that in this case, at least, the amyloid that is given to the patient also, the protein that's given to the patient determines the fate of the organs in the patient. I see that I'm getting, I would like to say, if I have a couple of more minutes, I would like to say something about type A amyloidosis as well, since there is now strong evidence that there is some association between wild type, which is type A amyloidosis, and different diseases in joints and ligaments. And one such disease is lumbar spinal stenosis, which is a common disease, and depending on squeezing on nerves coming out from the spinal cord. And associated with that is enlargement of ligaments here. And it has been shown and known for a quite long time that amyloid is quite common in this. And later it was shown that TTR is a protein that is deposited in this. And it's quite a lot of amyloid, actually. And this amyloid that of TTR origin is of type A, with fragments and full-length protein in a mixture. So we wondered if this amyloid can be the cause of lumbar spinal stenosis, and perhaps also be an early sign of systemic amyloidosis. So we looked at a big material of 250 patients undergoing surgery for lumbar spinal stenosis, and found actually amyloid in more than 1/3 of the material taken away from these patients. And in some patients actually also had severe degree of amyloidosis in this material. And what we also saw here is that if we look at females and males with a severe degree of amyloidosis, the males had a tendency to get this in younger age compared to the women. And what was also evident from this study is that the amyloid consisted of TTR, as is brown here in immunohistochemistry, and there was also another amyloid, at least one other amyloid, which is not labeled with antibodies to TTR. So there seems to be at least two different kinds of amyloid in these materials. You see it here too, quite a lot of other amyloid, and here TTR amyloidosis. And so one can answer, how can we put these findings together? We think that type A and type B fibrils are good examples of molecular strains, since they are same protein showing different shapes of the protein and of the fibrils and different diseases. And we also think that the properties of protein aggregates determine the tissue and clinical phenotypes. But how can we understand the changes in the ligaments? We think that it may be another protein, another amyloid that starts the whole thing. We can't prove that, of course, but it's working hypothesis. APOE1 has been shown to be present. Could be that, could be another protein as well. We are working on it. And by cross-seeding or secondary nucleation of TTR, it may be that ATTR seeds is formed in the ligaments, released to the blood, and then delivered to different organs, for example, the heart. And in some individuals, particularly men, then this gives rise to this severe heart disease in wild-type ATTR amyloidosis. And these patients, 250 patients that we studied, are now restudied 5 years later to see if any ATTR amyloidosis with cardiac deposits has developed. And we have some evidence that that is true, actually. Some acknowledgements here. I have to point particularly to Ole Sur, who has been the clinician working with these hereditary amyloidosis patients, and Per Eldhagen, who is the cardiologist working with these patients from a cardiological point of view, and Svante Berg, who is the surgeon that has coordinated the work with these samples from spinous stenosis. Okay, thank you very much for your attention, and I see that I have been 5 minutes late.
Thank you very much, Dr. Westermark, for a great talk. We already have a question in the Q&A folder. I can read it for you. You can also look at them. How is TTR
Can I look somewhere?
in the Q&A folder of your Zoom panel?
Q&A folder, yes.
Let me read it for you. This is from Velayudam. How is TTR exported from the liver cells?
Yeah, that, I'm not sure exactly. It's delivered as other proteins that are exported. It's delivered to the plasma, but I can't tell exactly. And it's actually delivered as a tetrameric protein. And it very soon also binds to retinol binding protein together with which is circulating in the plasma.
I have a very naive question. Does the TTR tetramer have any biological function?
Yes.
As a tetramer, not as a monomer.
As a tetramer, but not monomer. It's a transporter for thyroxine and also has an importance in transporting vitamin A through retinol binding protein. And it has also been suggested to have importance for anti-A beta aggregation in the brain.
Okay.
But I wouldn't be surprised if there are also other functions that are yet unknown, since it is difficult to understand what it does in the ligaments.
Okay, there's another question in the Q&A folder. Is the stenosis clearly connected to a specific TTR form?
It's a good question. I must say that I ask myself the same. I don't think it's very clearly connected to type A, but I'm not sure. It has not been studied, as far as I know, carefully in type B. But it occurs in many of the different mutations. But on the other hand, most mutations create type A fibrils.
Okay, we'll take two quick questions. Manjeet, you have a question. You raised your hand. Can you ask the question?
Yeah, we had a question here. How does TTR get important to cardiomyocytes?
Yeah,
And
go ahead.
I'm not sure that they are really. I should say that they were virtually intracellular in cardiomyocytes. The cardiomyocytes are complicated cells that have a system of T-tubules that are actually extensions from the extracellular space. So maybe these are virtually intracellular, but in reality in T-tubulus. But that's something that is yet to be studied.
Okay, Manjeet, if you're ready, you can unmute yourself and talk. Ask the question.
Yeah, so in type, a very interesting talk, thank you. In type A and type 2 fibril formation, it seems that in type A, there is a clear difference between the way both these fibrils are formed. In type A, the cleavage of N-term is necessary for the fibril formation to take place. Whereas in type B, you don't see that fragmentation happening. So the thing is that when the in type A, the C-term is fragmented, cleaved, it leads to the formation of fiber. Then when the fiber is formed, you said that the N-term is cleaved. So does that means that N-term is very flexible in nature and it stimulates the middle region to form fiber? To form fiber, and itself doesn't get incorporated into it. So the thing is that if we try to stabilize the flexibility of the N-terminal region, that might prevent this formation of type A fiber formation. Is that correct?
That's not so easy to respond to, actually, since I think the cryo-EM studies indicated that cleavage should not be important or necessary for formation of type A, since those results indicated rather that cleavage comes after formation of fibrils, but I don't think this is not completely settled yet. And I must say, my knowledge is not, my chemical knowledge is not good enough to respond to whether the flexibility of the N-terminal is more than, but I can say that the C-terminal part, starting at around position 50, is a very, very aggregating molecule, much more than the full length. So it's maybe that the N-terminal is much more flexible, but it's better to ask some chemist about this.
Okay, thank you, Manjeet. Let's take the last question here. Omar, can you ask a question?
Hi, can you hear me?
Yep.
Yep.
Thank you. Great, great seminar. My question is naive. I mean, do you think targeting TTR fibrils by specific antibody would be a good treatment for the disease?
Antibodies.
Antibodies, of course, immunotherapy. Yeah.
Yeah, there are trials on that, but I don't know how far these are gone. But actually, there are trials to make antibodies against TTR and to also other amyloid. So maybe it's.
Okay, thank you.
Thank you, Omar. Let's thank Dr. Westermark for the great talk, and we'll get back to the remaining questions at the end of the ZOOMinar. Now, I would like to invite Lucy Kamtamorian from Bodo to introduce the next speaker. Lucy, go ahead.
Yes, can you hear me?
Yep.
Okay, great, thank you. Yes, I am happy to welcome Marie Doumic. Marie got a PhD in mathematics in 2005. So she did a PhD in mathematics applied to physics and to laser plasma interactions. She was hired in INRIA in Paris in 2007 She got then a ERC starting grant, and she became the head of the research project MAMBA since last year. But since beginning this year, she created a new team in École Polytechnique next to Paris also, and the group name is MERGE for Mathematics for Evolution, Reproduction, Growth, and Emergence. Her domain research is mathematics, but it's applied to biology, and mainly she's working a lot in protein aggregation, and that's the reason why we invite Marie today, is to talk about how mathematics could help us to answer some question about protein aggregation. So please, Marie.
Thank you very much, Lucy, and thank you for inviting me. It's really an honor to speak here, especially after Per Westermark's fantastic talk. I would like to give you, let's say, 2 examples of what we mathematicians could hopefully bring to the biological understanding of protein and amyloid polymerization. So do you see correctly my slides now?
Yes.
Yes, it's okay?
Yes.
Full screen? Okay, perfect. So I just need, sorry, just a minute because I don't see them properly due to the other stuff. Okay, so polymerization, it's a ubiquitous mechanism, depolymerization, fragmentation. There are many applications. Of course, there are amyloid fibrils formation and degradation, but also you can imagine other kind of applications like actin filaments or mineral crushing. However, it happens that I really focused on amyloids till now for this kind of applications. And I would like to give you 2, I have written here 3 examples, but in reality, I will really focus on 2 of protein aggregation. And I say aggregation, but in reality, the experiments I will try to analyze mathematically are more linked to either fragmentation or depolymerization. And please, one thing more, don't hesitate to stop me if some things are not clear. I have to speak a little bit of mathematics, but I will try to be understandable for everybody. So let me first introduce the kind of experiments and of questions, biological questions coming from experiments that we have analyzed. So, sorry, yes, okay, sorry. So first, fragmentation. This experiment has been carried out by Weifeng Zhu at the University of Canterbury, and he agitated fibrils, amyloid fibrils, and you see from left to right what happens in the time course of the experiment. So here you are in vitro. There are only fibrils at the beginning of the experiment, so no interaction with monomers, for instance. And as time goes on, you see the fibrils breaking down into smaller pieces. And at the end, you see there are really many small pieces, whereas at the beginning, only very large ones. So he did that for 4 different proteins, beta-2M, alpha-synuclein, lysozyme, and beta-lactoglobulin. And the question is, what are the characteristics for this fragmentation? And this may be very important to understand, for instance, also the pathological abilities of each type of amyloids. How, from such an experiment, is it possible to estimate, for instance, where do the fibrils break down? Do they break preferentially at the edge, or do they break preferentially at their middle? And also, do large fibrils, long fibrils, break more or less than small fibrils? So all this is related to what is called the fragmentation equation. Here I show the experiments carried out on 3 other proteins. So first it was beta-2M, and then lysozyme, beta-lactoglobulin, and alpha-synuclein. And the question was also, are we able to compare for all these fibrils their characteristics, and can we link them to their pathological abilities? Second type of experiment, it's more a depolymerization experiment. So here again, we begin with polymers, more precisely to oligomers, relatively small polymers, ovine PRP oligomers. And here, the question now, it's no more fragmentation because the fibrils do not break in the middle. They really lose monomers at their edge. And the question is to estimate the depolymerization rate. So it's just an example to tell you, I will not speak more about this question. There are two questions: estimate the depolymerization rate and also the initial size distribution of fibrils. So it's just an example of what we could do. And third question, really more philosophical, or let's say qualitative. It's experiment carried out by Human Rezai at INRAE. And what he observes, and he and his team, is that it's the same experiment as in the slides just before, you see here. But now the fibrils do not only depolymerize, at some point they can also repolymerize by the addition of monomer addition. And what is especially interesting and surprising is that if you zoom, you see here, you can zoom on the pink zone. Here is a pink zone there. And then you zoom again in the small red zone. And what you observe are very fast oscillations. And this is puzzling because with our traditional depolymerization, polymerization mechanisms, it is impossible to observe such fast oscillations, which means that something else is happening, but we do not know what. So the question now was more or less, could you mathematicians propose us some chemical reactions that would explain such strange mechanisms? This will be the second part of the talk. Okay, so let's go back to the fragmentation experiment first. So our ideas are, how can we estimate the way the fibrils break down? So first of all, let's look at the experiment and write a model to understand what we really observe. So it's extremely simple. At different time points, as you have seen, we measure the sizes of fibrils. So we measure a sample of sizes of fibril x1 of Ti, etc., xn of Ti at different time points. How to relate this sample to some mathematical quantity? Well, we assume, and this is a big assumption, it is not true, of course, because the sample is not independent. Of course, each size of fibril depends on other ones, but we can prove mathematically that it behaves more or less like if they were independent. So independent, identically distributed, meaning that they all have the same size distribution, which has a given law, f of Tx. And here, U of Tx is the concentration. So we will focus now on the concentration of particles of size x at time t. Okay, so I will have to write some equations, but you can also only listen to what I explain and never mind if these equations do not mean anything to you. Okay, just it is, you imagine a concentration of particles, or it's as if you have infinitely many particles and you want to see what is the chance they break down. So then we can write an equation for this concentration of particles of size x. And you see, what do we see? We see that the evolution of number of polymers, so it is written mathematically here as du over dt at time t of size x, is equal to what? Well, there are some deaths, let's say, of polymers, which are the ones which break down. Breaking down, they leave the compartment u of tx. So there is this minus b of x u of tx, and b of x, what is it? It is precisely the breakage rate, the division rate. So what is important is that polymers of larger size can break down more or less as smaller ones. And that's why the division rate, the fragmentation rate b, depends on x. But they can also you can also have creation of particles of size X, and they are created out of larger fibrils that have broken down. So this is this integral term. So you have these larger particles of size X over Z, which break down and which give rise to polymers of size X according to a certain probability law, K0 of dz. Let's imagine, for instance, that you always break down in the middle. Then it means that polymers of size X can be created out of polymers of size 2X. Okay? So now we have a mathematical model. We know what we observe. We observe a kind of noisy version of the solution, the concentration of particles of size X at time t. We can assume that we observe that. And we want to estimate the parameters of the equation, which are precisely the fragmentation rate, B of X, and what we call the fragmentation kernel, K0 of dz. What is it? K0 is here to characterize the place where the fibril breaks down. If it is uniform, K0 will be equal to a constant to 1 on all the interval 0, 1. If it is in the middle, you will have a direct mass only saying, okay, I always divide in the middle of the fibrils. This equation has attracted much interest among mathematicians and physicists for a long time. But what is very nice for us also is that the questions coming out of amyloid fibrils are totally new and were not really looked at before. But just to tell you a little bit about this concentration, what is interesting, so I have written here a mathematical result, but I will now express it with words, okay? So never mind if you don't understand what is written on the slide, just listen. What is happening is that, so as you can imagine, the concentration of polymers U of Tx, the polymers break down more and more. So as they break down, you will, of course, end up with smaller and smaller particles. It is what you have seen at the beginning on the different snapshots I've showed you. Okay, but what is important is that they do not break down in any sense. What I mean is, as time goes on, given if you just reshape, if you look at the distribution of polymers, it happens that if you rescale it properly, it is what is written here, this rescaling t power 2 over gamma, g of t power 1 over gamma y. If you rescale it properly, it happens that there is a steady profile which appears. So this is really a very nice property liked by physicists and mathematicians. Many papers have tried to prove these results. But what interests us is that, okay, what can we say? How can we link? What we look at are the parameters of the division. So now I have done a big assumption, but this was done by Weifeng Zhu before me, also biologically. I assume that B of X is equal to X power gamma. And since, so it's like a power law, okay? So we need to have gamma strictly positive, but the question is, if gamma, for instance, is larger than 1, we expect it means that large fibrils will break down more than small fibrils comparatively. So I want to estimate gamma and alpha, and also this fragmentation kernel K0. And so the very first natural idea for mathematicians and for physicists was to say, okay, let's use this asymptotic behavior, try to observe it, to measure it, to estimate it. This is possible with what you have seen, since you just take a very large time distribution and see what you have, and try to obtain alpha and gamma out of that, okay? Just don't. Okay, so here, just some small simulations to give you a flavor of what is happening. So here you have the two opposite cases, either the uniform fragmentation kernel on the left-hand side. It means that the fibrils may break down at any place. And on the right-hand side, they break down exactly at the middle. And what you see is that as time goes on, there is a distribution which emerges. So here it is a rescaled quantity. Here you see the blue curve. It's more or less the asymptotic version. And here it is a green curve. And you see what is different when it is uniform. The size distribution is not zero around zero. It's like an exponential decay. Whereas if you take the fragmentation into two equally sized fibrils, then you have a one peak distribution. So it's relatively different. And it was our first idea when we began to discuss with Weifeng Zhu. It was, okay, let's use this mathematical property to estimate our fragmentation characteristics, alpha, gamma, and K0. And this idea was not so new. It was also used by some physicists, but I must say a long time ago, at a time when they didn't really have very good measurements. So I just cite a few papers here, just to say that it's really a relatively natural idea. And it's also similar ideas which are used for the cell division equation. So totally different application. However, cell division, there is also growth and fragmentation, division into equally sized, generally, daughter cells, or not equally sized for yeast, for instance. But still, and the difference is that you have growth plus fragmentation. In this experiment, I have only fragmentation, okay? So, sorry. So here, I have put some different asymptotic profiles according to different fragmentation kernel kappa, okay? So I rescaled it properly to have my correct steady profile appear. And then I see that what I said before, for fragmentation kernel, light blue, for instance, which is very strong at the edges, you see there is this light blue size distribution. On the contrary, for a fragmentation kernel, which is stronger in the middle, you have this dark blue curve, for instance, a one peak distribution. Okay, some examples of asymptotic behavior. Here now that we have the theory, let us go back to the experimental data that I showed you at the beginning, okay? 3 different types, 4 different types even of protein polymers. So here are the kind of data we have. So it is exactly the same thing. It is some snapshots, but as size distributions of polymers. So these are experimental data. So you see here at the top, you have 3 different proteins, and you have from left to right, the size distribution. On the top, on the back, it is simply a rescaling, okay, of the top. And you see that if you rescale properly, you see this kind of steady distribution which appears. Okay, so these are for the data. And now, before meeting us, Weifeng Zhu already has written, the very beginning of our collaboration was his paper with Sheena Radford in 2013, where he exactly did the same kind of assumptions of power law, for instance, for fragmentation, but he did it directly with a discrete model, and he compared the models by doing many simulations and trying to fit and to find the best fit one. What we proposed to him was to use the asymptotic behavior in order to have a computationally more efficient estimation, and also to use less data. So how did we do that? What we use, still in the mathematical properties, I will try not to enter too much in the details. We used, I will go directly, I would say here, to the slopes here, rather, okay? So what we did was to look only at one quantity over time. And this quantity was the mean length of the fibrils. So you see, you have all this distribution, size distribution over time, which is experimentally observed. We only take the mean, the average length of polymers over time. And we fit this curve with what we think mathematically is expected. And what is expected is a certain behavior over time as t power -1 over gamma, gamma being linked to the mathematical model. And you see, we have done this for the 4 different proteins. In reality, we did it, you see the onset on a log scale, because on a log scale, we see really well this power law appear. And you see, it is really natural to do the fit we did, since it is really well approximated by such a straight line. And the slope here provides us with the parameter gamma, which measures if the larger particles break down more or less than the smaller ones, okay? And you see, for the 4 cases, we thought, yes, okay, it seems reasonable to estimate such and to approximate the data by such a straight line. It is the only data we used, and this was sufficient to estimate at least alpha and gamma, not to estimate the fragmentation kernel K0, which characterizes where the fibrils break down. And only with this data, so here is the table of parameters that we obtained for each different types of proteins. I will comment them a little more afterwards. And using them, we were able to run simulations. So you see at the very first line, you have the experimental initial distribution of fibrils. We then run simulations with these parameters estimated on really simple, very few experimental data, only the mean size of polymers over time. And look how fit were really excellent. You see, on straight line, you see it compares the experimental data, which are light, and you see in darker color, you see the model fit. So at that point, we were really happy. We thought 2 things. First, it was really efficient because it is computationally extremely cheap. Second, it really means that our mathematical model is well fitted. Else, it is not possible with only 2 parameters, alpha and gamma, to fit as well so many curves. The drawback was that we had a little influence of the fragmentation kernel K0, but only a little one, less than what we would have expected. You see here, for instance, it is the simulations carried out with a fragmentation kernel uniform, and it's relatively good. And then just after, we did it with the fragmentation into 2 equally sized fibrils. And it is slightly less good. So we can have some conclusions, but not as many conclusions as we would have liked. What is, in reality, we have also analyzed more this problem. And in reality, it happens that the large time behavior, it's not really convenient. It's not enough to really characterize the fragmentation kernel. To characterize it, we need more. So to sum up our results, we have this table. And what we found, if I explain it very roughly, is that the one which is the less stable when smaller sizes, it's the alpha-synuclein. It is the less stable than beta-lactoglobulin, lysozyme, and beta-2 microglobulin. And this could be linked with its prion-like cytotoxicity, which has been proved in several biological papers. And because, you know, if small fibrils are less stable, they can break down and then give rise to still other ones by recruiting monomers, for instance. And probably it's linked to more toxicity. It is less stable compared to its width also, okay? So in a sense, we were happy because we were able to really estimate properly alpha and gamma, and it was totally in agreement with the previous results of the paper of 2013 that Wei-Feng Zhu did with Sheena Radford, but which was only on beta-2M. These new results were for 4 different proteins. But still, it was a bit a pity that we were not able to say much really quantitatively on this fragmentation kernel K0, because it is very important to know whether the fibrils break down in the middle or at the edges. So then we went back to simulations, and we first discovered, first thing, is that there is a time window where we have more difference, where we can see more difference in the size distributions with two different kernels. You see, we departed from two different kernels and we estimated the difference in the size distributions. And you see the p-value small means that it's really good. And here, left and right, we tested different kernels. Here you see the blue and green kernels are not that different, but still, if you depart with a peaked Gaussian, which is the curve in black, then during the time window between 0 and 2, let's say, or 0, at the beginning we have the same distributions, of course, you cannot distinguish. There is a way to select and to say, yes, the two distributions come from two different fragmentation kernels. So it was totally different kind of spirit. Instead of using the long time behavior, we want to use a short time behavior. And it is relatively logic because, so I would just like to show you that. If you think about it, if you have a short time, and if you depart, the best is to depart from fibrils having all the same size. The drawback is that it's, you will tell me, but probably it's not possible experimentally to depart with all fibrils having exactly the same size. But if you imagine that it is the case, and you wait for a time short enough so that part of them have divided once, a not negligible part of them have divided once, but a very small amount of these fibrils have divided twice. So you can neglect the ones which divided twice. And now you look only at the fibrils which have divided once, which are the smaller ones. Okay, it's easy. You depart, for instance, from fibrils having all the size 1. You don't look at the ones which have still size 1 after this small time. Then, of course, they are distributed exactly, that size distribution is exactly the fragmentation kernel that you are looking at. And it is expressed by these simulations where you see in red, large time. So in red, it is the asymptotic behavior. And in blue, you see exactly the fragmentation kernel. So it's 4 simulations with 4 different fragmentation kernel. And you see at the very first time, you see exactly the fragmentation kernel appear. So this is a kind of new work. So we have the first theoretical results which work very well, simulations which work very well. The problem is that having initial data, all having the same size, seems to be extremely difficult experimentally. So we are now developing methods to allow to depart from a distribution, size distribution of fibrils, not all of the same sizes. Okay, this is for the fragmentation question. If I'm allowed 5 minutes more, I would like to tell you about a totally different experiment, totally different because no fragmentation, but still it's depolymerization. So it's still smaller and smaller particles, which is a work done with, in collaboration with Human Rezai, biologist, and Mathieu Mézage, my PhD student, and Clemens Fellner, mathematician in Graz. And the previous I didn't say, but it's with Miguel Escobedo, Magali Tournu, mathematicians also, and Weifeng Zhu, of course. Okay, so let's look at this experiment that I have showed you at the very beginning. The first question when we presented this was, of course, to say, well, are you certain that there are oscillations? Are you certain it is not noise? You see here A and B, A, B, and C, it is at the beginning. And at the end, of course, it's only noise, okay? D and E, it's only noise. So we first built a test to say yes or no, there is or there is not oscillations, okay? And we see that, of course, at the beginning, there are clearly oscillations. And second step is, what kind of mathematical models could explain that? So compared to the previous work, it's much more phenomenological. The idea is really to try and to propose some chemical reactions. I do not say that that these reactions are the real ones which really happen. I simply say they may be the good ones. And now I let you, Ruk, to say whether it's truly confirmed by experiments or not. First, we departed naturally by the standard depolymerization model, okay? The most natural model for polymerization, depolymerization is written here. It simply says that Ci, polymers of size I, depolymerize, giving rise to polymers of size Ci-1 plus monomers C1, okay? And we can add polymerization, even if it's negligible. C1 plus Ci gives Ci plus 1. This is called the Becker-Döring system. There are many rugs also on this system, but it has a property which is called detailed balance and which gives you a result saying no oscillations are possible. Now, which simplest reaction system may give rise to oscillations? One possible answer is the so-called Ivanova or Belousov-Zabotinsky model, very much simplified, okay? So it's scissor paper game also. If you have V plus W gives rise to 2W, W plus M gives rise to 2M, and M plus V gives rise to 2V, then it really gives very coherent oscillations, unsustained oscillations, never vanishing. So our idea was to mix these two kinds of systems. And here is what we proposed. So it's exactly the same Becker-Döring system. W plus Ci gives Ci plus 1, polymerization. But instead of having the normal depolymerization, which is Ci plus gives Ci minus 1 plus V, we proposed a catalytic depolymerization, meaning that Ci needs to react with V, which attaches maybe a little, and which then detaches with another V. So it's Ci plus V gives Ci minus 1 plus 2V. So we wrote this system. I was already a bit too long, so I will not detail the analysis and why it is oscillating, but here are the kind of numerical simulations we get. So it oscillates extremely well, almost too well. And of course, this catalytic depolymerization, we discussed a lot with Human Rezai. I would be really happy to have your opinion on that reaction. It can only be some, I would say, only, it cannot be the main depolymerization reason, but it could be a kind of small correcting reaction. And this is what we proposed here. We say, okay, let's add in our system the standard depolymerization reaction. Ci gives Ci minus 1 plus W. Afterwards, you see we have these two monomer species, W and V, which react together. And then you see that qualitatively, at least, the kind of numerical simulations we obtain really look like the very first experimental results I have showed you. So as a conclusion, I would simply comment a little bit saying that we have to adapt what we are looking at, asymptotic theory, as I showed you at the beginning, to estimate the parameters of the fragmentation. It's a beautiful theory. It's partly useful, but only partly. So we cannot use it systematically. And now there is this idea of using the short time measurements. So we really need to adapt our mathematical theory to first to what we want to estimate. And also it would be nice to adapt the experimental measurements to the question we have. And also math could help to propose mechanisms, even if we are not able to validate them, of course. And for this, it's one more the work of biologists. And I really enjoyed very much all this back and forth between math and biology, and I hope to continue a long time still. Well, thank you very much. I don't know if I should stop the sharing of the screen.
Thank you very much, Marie. It was a really nice talk. It was a bit difficult, I think, but it's really nice to see how a mathematician could help us to better define our model and the dynamic of our amyloid proteins. So I think we have one question. Yes, from Ashutosh Tiwari. He say, you can read it also in the Q&R, but he say, a very interesting talk. Have you tried to predict some aggregation properties or depolymerization properties which have inspired experimental work where the data corroborates the mathematical prediction?
So I would say that what I showed first with the fragmentation equation, with the estimation of alpha and gamma, really corroborates, because you see, if I go back to this data, here, we were really happy because we used only very part of the data, of the experimental data, only the average size of fibrils over time. And it was sufficient to estimate properly all the size distribution of data, so really much richer ones. So in this case, our mathematical model really fits the data that we didn't, and we didn't use the data for all the data, only part of the data. You see, so in a sense, part of the data was used to predict the rest. So it really corroborates the fragmentation model, which is also interesting because we know that for all this, and clearly Per Westermark's talk showed it, there is a wide variety of fibril structures. And despite this variety, a very simple model, even if it's simple in the sense that I assume that all the fibrils, given their length, behave similarly, which it is sufficient to explain this. Now, what I would like very much would be to estimate really better the fragmentation kernel. The next step would be to obtain, if possible, experiments where we depart from a monodisperse size distribution of fibrils, meaning, is it possible to depart from fibrils having more or less all the same sizes? This would be really nice to know how they break down. Now, concerning the second part of the talk, what is Human Rezai seems to really believe in this catalytic depolymerization. I think he begins to have some experimental evidence, but it's not yet fully validated. So I would love that it would be, or on the contrary, that it would be contradicted, you see, in any sense. But it's not fully the case yet. I'm not sure I've answered the question, but yeah.
There is two questions from Philippe Neudecker. The first one is, why is it that you can neglect growth? Don't you have monomers that can attach? So
thank you. I should have told it. Yes, for the first experiment, so the experiments of fibril fragmentation with Weifeng Zhu, the concentration is really too low, so that he really experimentally proved that polymerization is negligible. For the second talk, on the contrary, we did take into account polymerization, and it is absolutely necessary. If we forget about polymerization, then we cannot have these oscillations, at least not with our models, maybe with other ones, but not with, I think it's not possible. Now, so,
yeah, Philippe has another question. Why should the kernel be inhomogeneous if you have no mechanical strain? Can't molecules dissociate everywhere?
I am not certain I understand what you call inhomogeneous for the kernel. Yes, the molecules can dissociate everywhere. And this means a uniform fragmentation kernel, what I called uniform. But uniform means that equal probability at any place of the fibril. And this is experimentally not really true. You see, if I look at here, at the sum up.
Yeah. Sorry, Marie, but Philippe just said that inhomogeneous for non-constant, not constant. In the sense of non-constant.
Okay. Then, because it may happen that you do have mechanical properties of the fibrils. Weifeng Zhu proposed a kernel which is stronger in the middle due to some physical law that I have forgotten. But you see, there is agitation. So it's not just, of course, if nothing happens, then you could break anywhere. But there is agitation, so the fibrils move. And it's possible that they are, for instance, weaker at the edge, or on the contrary, weaker at the middle, or something like that. There are some laws which give you some power law, for instance, for the kernel. And what we proved, but for the kernel results, it's where we are less confident than for the fragmentation rate, is that, for instance, for alpha-synuclein, they break more in the middle, whereas for beta-2 microglobulin, it's more uniform. Okay.
Thank you, Marie. There is maybe a question. Someone raise hand. Sorry, it's Manjet Kumar. So I don't know if it was for the previous talk or if it is for this talk. So Manjet, if you hear me, you can you switch on your microphone? I will try to give you the sound. Or could you type your question in the Q&A, please?
Maybe it was for the previous talk, yeah.
I think so, but I'm not sure.
Are there a question too?
There is no more questions. Philippe just say, okay, so there is no mechanical strain. Thanks.
There's one question in the Q&A.
Yeah, sorry. It's for, I think it's for Per.
Yeah.
No. Yes, it's for Per. It's does COVID-19 spike protein bind to TTR and facilitate the amyloidosis?
Can I see?
Yes, I think you can see it, Per.
Do you want me to stop the?
You can, Marie, I think. Do you see, Per, the question?
I can't see any question.
You want me to say it
see
again?
the bottommost question in the Q&A folder. I can read it for you. So, great talk. Does COVID-19 spike protein bind to TTR and facilitate the amyloidosis?
Growth. Let's see. There, fine. That's COVID-19. The question regarding COVID-19, spike protein, I have no idea. I don't think there is any experiments on that. I'm not what I'm aware of, at least. When it comes to TTR amyloidosis, I think it has been suggested for some other amyloid.
There's another question from Ada from Munich. Do results from domino transplantation of livers from patients treated with tafamidis exist? In the Q&A folder, there's in the middle somewhere you see Capernuto, the last name, Ada Capernuto.
You mean I can treat patients? Yeah, sure. You mean the recipients of livers?
Mm-hmm.
See, do results from abdominal transplantation of livers from patients treated with tafamidis exist? Yes, they do. And I'm not an expert on that, so I don't know the results of that, but certainly it's they are treated with tafamidis.
So do they differ from the results with the non-treated patients?
Not as far as I know, but I'm not an expert on that.
Okay. I'm not sure if you covered this. Do the types A and B cross-seed each other?
We have not tried this experimentally, and you mean as in vitro studies, that should be possible, but I don't think that we haven't done it, and I haven't seen any such studies either.
If they cross-seed, do you expect any biological significance? They do coexist, right? So why can't they cross-seed? It's possible. I don't know. What would be the significance?
If it should have any clinical,
yeah,
I guess so. But on the other hand, I don't know. I think we have to see the results first. It could, of course, have clinical importance, but I don't know.
I have a question
Go
for
ahead,
Marie.
yeah.
If we are doing experimental, it's what we are doing us, and we want to use mathematics to analyze our data. Which kind of experiment would be the best for you to do? And do we need to do only one experiment like THT or AFM, or do we need to do more than one type of experiment? What is the best for you to try to correlate mathematics with what we have in our tubes?
Thank you for your question. I would say that THT would, for instance, measure only an average polymerized mass over time. Am I correct?
Yes.
And this is relatively difficult to use mathematically. It's the second part of the talk, for instance. And that's also a reason why it was not THT, it was static light scattering, but it's the same kind of data, which means you only measure one quantity over time. And so it's difficult because many different mathematical models could give rise to the same kind of experimental data. So I would say that AFM is really, if it's possible to have several time points, AFM gives rise to much more information because we have then access to a size distribution of fibrils. Okay. And even size distribution, it remains difficult because you don't know, at least I think for the moment, it's not possible to know which fibril has breaking down into two pieces or which monomer has attached to a given polymer, you see. But still, it's possible with this indirect estimation, with the modeling we have, the techniques we have, to estimate the features, the characteristic features better. So I would say that size distributions are really one key.
So it will be better to do AFM than to do THT.
I would say so, but you know, yeah, it's
no, but we have to know because we are doing experiment and we are giving you the experimental data to try to fit. So, okay.
I think it's better to say quantitative things, not only qualitative.
Okay, thank you. I think Danilo has a question.
Yeah. Hello, everyone. Sorry for connecting late, but I was a bit confused for the new time, so I was really wrong. I was trying to connect on a wrong time. So I'm not familiar with the field, of course, but I have a question for Marie. Do you think it's possible to model by means of your equations the cross-seeding of different kinds of polymers?
Oh, so yes and no. Yes, it's possible. Clearly, it's a matter of time and understanding of what is exactly cross-seeding, and it would be certainly really interesting. So yes. Why? No, because I have to say, to stay modest due to the fact that if it's really too complex, if we put too many things in the model, then it will be possible to say almost anything. So it's more or less linked to my answer to Lucy. So if you mean cross-seeding, how many different species do you have?
Let's say two.
If it's only two, if it's only two, I think then we can really say something.
Two from different proteins.
20, or then I could write anything, but it will be maybe more difficult to say something really quantitative. But yes, two, I think clearly it's possible.
Okay, that's fine.
It's always possible. The problem is if the model is too complicated, then
yes, I understand.
points and I fit an elephant. And
Okay,
Okay.
let's thank both the speakers, Marie and Per, for the great talks. Thank you all very much for joining us. See you next Monday.